In this activity, we're going to model meiosis using paper chromosomes, just like we modeled mitosis earlier. This time, however, the goal is different. Meiosis reduces the chromosome number by half and produces genetically unique cells. As you follow along, you'll watch homologous chromosomes pair, exchange genetic material, and then separate through 2 rounds of division. If you played along with the mock mitosis, feel free to do the same with meiosis. Doing the movements yourself helps you learn through muscle memory. You need the same materials as mock mitosis, except you will need an additional piece of paper, 2 more pieces of yarn, scissors, and tape. Meiosis is a special type of cell division used to produce gametes. It cuts the chromosome number in half and introduces genetic variation. This is important to do since male and female gametes, which are haploid, come together in sexual reproduction to form a diploid cell again. Before we begin, pause the video and write your prediction in your lab notebook. Think about what happens during fertilization when 2 gametes join. Resume when you're ready. This is a diploid cell entering meiosis. This would be a specialized diploid cell called spore mother cells, which are located in the reproductive organs. This would be pollen mother cells in the male anthers of a flower or megaspore mother cells in the female ovules of a flower. During the S phase of interphase, the chromosomes replicate. Each colored paper strip represents 1 chromosome. When you clip 2 strips of the same color together, you are modeling sister chromatids joined at the centromere. Because you have 2 different colors, you are also modeling homologous chromosome pairs, 1 chromosome from each parent. At this point, the cell contains duplicated homologous chromosomes, but they are still inside 1 nucleus and have not yet paired or separated. In prophase 1, homologous chromosomes move toward each other and pair up in a process called synapsis. When you bring the same colored chromosome pairs together side by side, you are forming a tetrad, 4 chromatids aligned together. Crossing over occurs when sections of chromatids exchange DNA. By cutting the ends of 2 chromatids and swapping the pieces, you are modeling genetic recombination. I did this crossing over in 2 places, since crossing over doesn't just happen in 1 place on the chromosomes, but in many places. This creates new combinations of genes that did not exist in either parent chromosome. During metaphase 1, entire homologous pairs line up at the center of the cell. This is different from mitosis, where individual chromosomes line up. Homologous chromosomes randomly orient themselves along the metaphase plate using spindle fibers. This random orientation leads to independent assortment, another major source of genetic variation. Since this is more difficult to see with only 1 tetrad, I added a diagram that shows random assortment of 2 tetrads. In anaphase 1, homologous chromosomes separate and move to opposite poles, but the sister chromatids remain attached. When you move 1 full paper-clipped chromosome of each color to opposite sides, you are modeling the reduction of chromosome number. At this point, each future cell will receive only 1 chromosome from each homologous pair. After the homologous chromosomes have separated, 2 cells form. Each cell now has half the original chromosome number, but each chromosome still consists of 2 sister chromatids. This is why meiosis 1 is called the reduction division. The nuclear envelopes may briefly reform and the cytoplasm divides through the formation of a cell plate. This is shown by placing each on its own paper. Meiosis 2 begins in each of the 2 haploid cells. In prophase 2, the nuclear envelope breaks down again and spindle fibers reform. In metaphase 2, now the chromosomes line up individually just as they did in mitosis. When you place each paper-clipped chromosome at the center of each new cell, you are modeling this alignment. Now the centromeres split and sister chromatids separate. When you remove the paper clips and pull the chromatids apart, you are modeling how each chromatid becomes an independent chromosome. This step ensures that each final cell receives only 1 copy of each chromosome. After the second division, 4 haploid cells are formed. Nuclear envelopes reform around 4 sets of chromosomes. Because of the crossing over and independent assortment, none of these cells are genetically identical to each other or to the original parent cell. Now that you've completed the meiosis model, compare the final outcome to what you saw in mitosis. In mitosis, the goal is to make identical copies. In meiosis, the goal is something very different. Consider both the number of chromosomes in each cell and whether the cells are genetically the same or different from one another. Pause the video and write your answer before continuing. During prophase 1, homologous chromosomes pair and exchange segments of DNA. Think about what happens to gene combinations when pieces of chromatids are swapped. How does this process create new combinations that were not present in either parent? Pause the video and explain how crossing over increases genetic variation. Remember that DNA replication already occurred before meiosis 1. If replication happened again before meiosis 2, think about what would happen to the chromosome number in the final cells. Would the cells still be haploid? Pause the video and explain why skipping DNA replication is necessary. In metaphase 1, whole homologous chromosomes line up as pairs. In metaphase 2, individual chromosomes line up, similar to mitosis. Think about what is being separated in each division, homologs versus sister chromatids. Pause the video and describe the difference in alignment. Not all stages of meiosis create variation. Think back to where chromosomes exchange DNA and where they line up in random orientations. Which phases introduce new gene combinations and which simply separate existing ones? Pause the video and identify the stages that generate genetic diversity. This model shows how meiosis creates genetic diversity while maintaining chromosome number across generations. Structure and chromosome behavior during each phase ensure both reduction and variation, key to plant reproduction and evolution.